Hello, and thank you for joining us today for this webinar, Progressing Drug Therapies to Market, the Criticality of Analytical Testing Robustness, which is brought to you by Scrip and sponsored by Cambrex. My name is Andrew Warmington. I'm the manufacturing editor at Sightline, and I'll be your moderator today. Before we jump into the presentation, let's go over some housekeeping items. First, if at any time you're having technical difficulties, click on the Test Your Connection button, and this will take you to a connection checker and FAQs page. If you're experiencing any technical issues, there's also a Chat Now function to contact the support team for assistance. If you want to enlarge the presentation, just click the Maximize icon or drag the lower right-hand corner of the presentation window. There'll be a Q&A at the end of the presentation, so if you have questions, we want to hear from you. To ask our presenters a question, simply type it into the question window at the bottom of your screen and then hit Submit. We'll be answering as many questions as possible during the Q&A session, and if there are any extras we don't have time for, we'll try to answer you after the webinar via email. And because we know you'll want to listen to this webinar again, it's being recorded and will be available to view on demand in about 48 hours. I'd like now to introduce our speakers for today's session. First, Tracy Milburn, who is Director of Analytical Services at Cambrex. Tracy's 25 plus years in analytical chemistry span early stage development through to product manufacturing. Her experiences range from drug substance and finished product analytical method development through to quality control in support of commercial manufacturing. She has supported IND, ANDA, NDA, and other regulatory submissions via designing support studies, conducting testings, and authoring support documentation. And Daniel Kirschner is Executive Director of Analytical and Biopharmaceutical Services at Cambrex. Daniel holds a PhD in bioanalytical chemistry and has been a scientific leader for 15 years, with experience in both biotechs and CDMOs. He's worked at all stages of clinical development, from pre-IND through to registration, and is the author of multiple manuscripts in analytical and organic chemistry. Daniel holds several patents, and at Cambrex, he's responsible for all aspects of CMC analytical support. And with that, I'd like to hand things over. Tracy and Daniel, the floor is yours.
Good morning, everyone. So we'll jump right in a little bit to talk about the CMC development from your early stage to late stage programs. I'll be talking about the phase 1, phase 2 cycle of events, and Daniel will take over for the transition from the phase 1, phase 2 into phase 3 commercial to move you forward to complete your drug product development. So let's talk a little bit to start out about your goals of your phase 1 and phase 2 drug substance development. Initial discovery and route scanning, right? This is the time that you're wanting to find out if your path to a molecule is real, something that you have maybe a development route for, a medchem route for, to see if you'd like to have an application that will treat a drug. So you're looking at a gram scale route. This is your first pass at finding a scalable manufacturing route. It's your first pass at identifying your raw materials, at deciding whether or not your starting materials are going to need to be custom-made materials, or if they're going to be materials that you can buy off the shelf. What do you need for those materials? Do you need to confirm identity? Do you need structural confirmation? This is your phase where you're trying to really nail down what it is that you're looking for just to get to your molecule. This is where you want to generate impurities if you can, and you want to get your intermediates identified between the steps of your manufacturing process, all to get to that place where you're going to get your initial characterization of your molecule. This pass 2, you want to put on stability if you can. If you get enough material, you want to see what happens in 6 to 12 months. If it's enough material to get at least an impurity analysis to know where you're going from. Next step for you is probably a 1/10 scale production run, your scalable run, to see whether or not you're going to be able to make material to get into a clinic. From this, you're gonna probably want to generate your reference material, maybe a couple of reference materials just to make sure that you have enough. This is where you want to do stability testing to cover a clinical study, usually your GLP tox-lot. To cover the length of the study or longer, this will help you set your shelf life. So if you can get 12 to 24 months out of this material, that'll help you long-term determine what your shelf life will be and to start your impurity profile development. And then finally, your clinical lot. This is usually probably a 5 gram to 10 gram scale lot that will go into your first in human. This will be your first ICH stability program and will help you expand your shelf life. So what I'm talking about today is the first part of your whole journey. The initial phase 1, phase 2, you're here in the process. This is the fun part for me. And I know that probably Daniel will disagree as he gets a little bit into his presentation piece of this, but this is the fun part. So this is where you're finding stuff out. You're learning about your molecule. You're learning what it's capable of. You're learning where it can go and what it can do. So for your initial filing, you're headed to your IND at this point. And what you really need to keep in mind at this stage is the I in IND is the most important part. This is your investigational new drug. So you don't know anything right now. A lot of people at this point have some misconceptions for their initial filing that make it very difficult to get where you need to go along the road. A lot of people think that initial filing has to be perfect. You've got to go in with methods in final form, that you know your impurity profile is all known and identified. But the reality is not that. There's no such thing as a perfect filing. You want it to be good enough to get through the agency. You want it submitted. You want them to accept it, knowing that your methods will evolve over the life cycle of the product. Things are going to change, and they should. You're in your first pass. Your molecule, your methods, your whole process is just in a kind of an infant stage. So you should expect that there will be development and improvement over the life cycle of your material as you move forward. You're going to see additional impurities be generated over the life cycle. So as you're starting these initial stability studies, what you think you know about your molecule and how it decomposes, what it degrades into, we don't really know yet. So new impurities should be inspected. Your profile is going to change. Your process is probably going to change. Your first scalable process to get you to that clinical lot is not the process you're going to go into your validation lots for in your phase 3 and commercial development. Toxicology studies may need to be reperformed as your impurity profile changes or evolves. So you should expect to repeat some of the work that you do at early stage. You should expect to repeat that as you move through the life cycle, which leads to possibly reevaluation and revalidation of methods throughout the entire course of the life cycle of the material, even past the commercial stage. So don't be surprised if something pops up and you find out you need to change a method, particularly your related substances. So let's focus on that a little bit. So the method life cycle is an interesting thing depending on the method that you're dealing with. You're looking at method verification for USP methods. You're going to do that throughout the life of your product. Simple tests, appearance testing, compendial testing, micro, all of that stuff only needs to be verified. Method qualification, phase 1, phase 2, that's where we sit right now in your process. Those methods need to be qualified, which is just really a short version of the validation. You're really executing everything in the validation of like an assay and impurities method, with the exception of maybe robustness. You're going to look at all these things. You're going to look at linearity, specificity, precision, accuracy, your LOD, LOQ. You're going to look at all of that stuff, and you want to do that. You want to know what your method is capable of. You're just not going quite the full length of validation, which comes at your phase 3 and commercial product, which is not what I'm talking about today. So let's talk about that early stage development. What are we looking for? We want to know the methods. At this stage, we know the methods do not need to be qualified or validated, only scientifically sound or fit for purpose. Sorry about that. What does that mean? We know that they're good. The separation for your degradants is sufficient. Maybe not perfect. Maybe you don't have everything teased out. You know where your starting materials and intermediates are in your chromatography, so you know that they're separated sufficiently to allow accurate determination. Same with your unknowns. Linearity is proven, so you've gone the length of knowing that you have linearity from your LOQ all the way to about 120% of what your assay value is expected to be. So you can see that and you can prove that. LOD and LOQ are appropriate for the level, which is usually 0.02 and 0.05% area respectively. And you don't have such a noisy baseline that is jeopardized. The accuracy of the method, you do want to do some spiking at this stage to know that your method can recover at 100 to 120% of your target value. You're looking for probably recovery levels from 90 to 110%. All of this for your initial method is sufficient. As your method improves and as you move forward with the process, you're going to tighten up some of these parameters as you move towards validation. So let's take a real quick look at a project that we worked on recently here. So this project for us was a method that was handed to us from early stage development. This was initial route scouting. This was the method that they had brought to us for their molecule, thinking that it would be sufficient to move forward. And to be quite honest, it is sufficient to move forward. It's not great. So what are we seeing from this? Separation is sufficient, but not perfect. So we're looking at these peaks starting ahead of the main peak. We can separate that out. We can see that it's a separate peak. We know that there's something there, and it can be integrated from the main peak, but it's not great. The resolution is less than 2. We don't like that so much. We'd like to see that a little bit better. Linearity is proven. We do know that the main component is linear across the range that we need to see. The LOD and LOQ are appropriate. We can see very low on this method, and the baseline looks good. So we know we can see where we need to see to be able to report accurately. So it's an okay method. It's not the best. Our recoveries are good. We're looking really at very good recoveries at 90 to 99 to 100% for each of the levels that we're spiking. So it's okay. Not the best method we've ever seen, but it's okay. So if we need to get stuff out the door tomorrow and we need to be able to provide a C of A for it, that gives us a good idea of what the purity of this particular molecule is, we're okay. We can use this method. We don't really want to move forward with it, so we're going to look at some ways to optimize it. So we'll need to pull this out. We'll need to look at some different types of columns, maybe. Look at the gradient in the mobile phases, perhaps. Maybe the first pass at this method was pretty quick, and they were just trying to get separation to know where we're at. Column conditions may be adjusted, temperature, all of that kind of stuff. So we did that. We did some optimization of the method, and we ended up with this as our final method. So the method was improved by changing the column. We went from a larger bore column to a shorter, smaller bore to get a little bit more definition in the peaks that were coming off. We adjusted the gradient percentage and the hold times. So we changed the rate at which the peaks are eluting off of that column. And we included THF in the modifier, in the modifier for the mobile phase to allow for better separation. By doing that, we were able to get full separation of these peaks. And I apologize, that's a blurry little picture there. But we were able to pull these peaks out. And indeed, that peak prior to the main component ended up being 2 peaks that were separated from the main component with almost baseline separation. And we were selected this method to progress from into initial forced degradation and qualification of the method to support the tox stability a lot. I would point out at this stage that this is the final method that we chose. However, this is not the final method that will probably end up living the lifetime with this molecule. You can see, even though the chromatography is better for this material, it's still not probably where we want to be for a commercial program. So to get us through the first phase, to get us into a commercial lot and get through that first stability program, this method is workable. But still, you can tell just based on the separation here, we do want to pull that out a little bit more, and we'll continue to do work on that to make sure that we can adequately see what we need to see and know that we're getting everything we need to know. So from this, we're going to move into forced degradation. I did want to talk a little bit about forced degradation as we move forward here, because I think there are a couple of there's different philosophies at this stage. Typically, the forced degradation that we're looking at right now is specifically to determine the degradation pathways. Provide information regarding the primary challenge stability conditions. So where does it break down? What's causing it to break down, and what does it look like its storage condition needs to be? We want to know that the method is capable. It may not be fully optimized, but it is capable of seeing breakdown compounds, that we're getting full separation. What we don't want to look for at forced degradation at this stage is mass balance. So a lot of people want to see mass balance at this stage, but it's not really the point of the forced deg at your early stage. Your phase 1, phase 2 forced degradation studies will be really to make sure that you have separation and that you understand how your molecule breaks down. We may want to identify impurities, but at this stage, we're really not looking to identify specific impurities. We are still, again, just looking for the degradation pathways and to know where our problem spots are going to be for the molecule. If something pops up during forced deg that we want to take a look at and want to maybe do that ID on, we can do that, but it's really not an expectation for the early stage forced degradation. So we're looking at all your ICH conditions. We are looking at heat, oxidation, basic conditions, acidic conditions, light exposure, all of those things we want to take a look at. And again, that'll feed both your shelf life program and understanding of the molecule. Understand that you will do forced deg multiple times throughout the course of your development. And again, for different purposes. Each time you change the method, you're going to want to do a quick forced deg study to make sure that whatever you've changed in the method is still seeing what it needs to see. When you get to validating the method, you're going to want to repeat a fuller, a more robust forced degradation program that does include mass balance. So keep in mind that as your product develops, you're going to continue this. Probably 3 or 4 times you'll see forced deg cross your path. Stability studies, again, let's touch on that just real quick. Types of stability studies at this phase. The goals of the stability studies at this phase. The goal really is to set your specification, right? To understand your molecule and to know what you're going to move into your later phase studies with your spec stat at. These studies will feed a lot of information for you. The more you can get out of these studies, the more robust you can be with your later stage submissions. To get into the IND, you really need 1 lot, 1 month of data, and to continue submitting that data as it's collected. Keep an eye on your data at early stage. You'll be able to use that to extend your shelf life as long as your product is stable. So that's really the goal for the early stage studies, is to understand your molecule and get yourself to a place where you understand also what your shelf life is going to be. So pilot stability. This is not it's not a required thing that you do for your IND submission, but it is really, really super helpful because you'll get this material way sooner than you're going to get your ICH stability lot. It will help you determine paths forward for that ICH stability lot. It'll get you storage conditions for the bulk material. It will help mitigate any surprises along the way. The worst thing you can possibly do is to get 2 or 3 months into your ICH stability program and find out that it's not stable at your primary storage condition, or that you're seeing a degradation product that you didn't expect to see. If you have the opportunity and the ability to put your pilot lot on stability, definitely do that. The tox-lot study material. So this is your bigger batch. This is your probably 10th scale batch. You definitely need to have this on stability. You do need to have this on stability minimally to cover the initiation of the study and the end of the study. Hopefully you've run that pilot stability so you know what conditions you're going to put your material up at, and you know how to store that material. You're looking at degradation products for the most part. A lot of times it's difficult to decide what you want to put up, what testing you want to do for your initial tox-lot stability, but primarily you want to look at those impurities because you're also using this lot to qualify those impurities. This one, to me, at the early stage, the GLP tox-lot is almost the most important study you can do because you can use this also to set your shelf life. It can be your initial filing stability program. You can move forward with this one in a lot of ways that you may be able to get ahead of that ICH stability if you start with that tox material. And then finally, the GMP tox or the ICH stability lot. This is your 5 to 10 kg lot that you're doing your first in human studies with. So by now you should, if you've done the preliminary stability programs, you know your storage conditions of what you're going to suggest for commercial storage. You have an idea of what the shelf life of your product should be. You know the degradation pathways for the molecule as it exists right now, and you know where to see those impurities in your impurity method. Typically, this study is a 24-month study. We do usually recommend that you put more material into that chamber than will cover the study, just in case you want to extend the study or add a time point, or there's investigational testing. But minimally 24 months will get you where you need to be with this lot. So I'm going to hand it over to Daniel now. and he'll present the transitioning it from this phase into a later phase program. Daniel, you ready?
Yeah, 1 second here. Let me share my screen. All right, I hope everybody can see my screen. So, morning, everybody. So as we transition methods from early phase to late phase, this is really an exciting time. The transition to late phase is a journey, and I put a picture here of a windy road because as the program starts to evolve, you're going to be learning a lot more about this program. One of the reasons this is so exciting is the clinical readout that has come from this drug. So at the stage when you get into about middle of phase 2B, you have all of your initial drug safety studies. Those are usually done in your clinical phase 1, but by the time you do your phase 2B, you know not only is the drug safe, but the drug has potency against its primary target. And that's a clear indicator that this drug is likely going to be a viable candidate for a phase 3 clinical trial. You also have more clear understanding as you move into late phase of the dosage form and the strength that you're going to be moving forward with. So it's an exciting time in the clinical side, but it's also an exciting time in chemistry, manufacturing, and controls. As we move into late phase, we've now produced more batches to supply not only our phase 1, but our phase 2 material. And we gained a lot of knowledge. That knowledge that we've gained isn't just in the analytical, it's also in the manufacture as well. So our process chemistry team has been hard at work to understand how the process can be tuned and optimized. They've looked at purge of specific types of impurities. So perhaps they've redeveloped a new recrystallization route to improve the purity of the API. They've improved some of the yields, and they've worked through difficult routes. As they've done that, they've been working closely with the analytical chemistry team that has been hard at work identifying our primary GLP tox impurities. Those are those impurities that would have cropped up in your initial tox batch. Those impurities will get identified in the phase 2 clinical studies and moving into late phase. Also, our QC team is starting to have a clear and better understanding of the stability of the API and drug product as we move out of phase 2 and getting ready for phase 3. We now have usually 2 to 3 years of ICH stability for our drug substance and our drug product, and we feel pretty comfortable at this stage for how the drug behaves, and we understand some of the key degradants, how they're forming, whether moisture uptake in our product is a problem. We can tune our packaging configuration if we find those kinds of situations cropping up. Phase appropriate methods at this stage, they're functional. They're working for their intended use. They're able to look for key degradants and impurities, but the analytical methods will need to be put to further scrutiny. So this is really an exciting time at this stage of development. The change to late phase, it also brings fresh focus areas to our CMC development program. To navigate that change, ICH has some requirements, and the FDA has some guidelines to help navigate. Some of the key things that we're going to be looking for as we look at building a control strategy for phase 3 for our drug is the process scale-up. We need to be mindful that process scale-up, we're going to be moving from maybe tens of kilos up to hundreds of kilos of API. Our process scale-up, it can result in new critical quality attributes. So I use that term CQAs shown here, but the critical quality attributes is a specific attribute about that product that we have to control. Some critical quality attributes we control by specifications, some we control by the manufacturing process itself. And it's going to be new CQAs that are developed that we need to have a clear understanding of. Method lifecycle management, where is our method at? That's going to be addressed. We're going to either have to look at certain methods that need to be optimized, or we're going to be doing some redevelopment work. We also have to have a clear definition and control of the inputs that go into our manufacturing process. We've been very focused on the API, making sure we can generate quality API and drug product. We're needing to take a couple steps back and start looking at the earlier stages of our process for registration starting materials. We're going to be looking to identify all of our ICH Q3A impurities to understand not only what they are, but where they originate. And are they originate one step back from the API, 2 steps, or all the way from the starting materials? So we need to understand their origin, the fate of those impurities, and how they're purged through the manufacturing process. A key part in transitioning to late phase is the synthesis and control of reference standards. When we start out in early phase, there's not very many. As we move into late phase, the requirements increase significantly. We're also going to be looking at other attributes like genotoxic impurities and nitrosamines. We're going to be looking at our drug product packaging and doing extractables and leachable studies. A big point here is development of robust analytical methods. So for the purpose of the webinar, there's a lot going on in the transition into late phase. We're going to focus on registration starting materials, reference standards, and development of robust methods. As I mentioned, to navigate the change, ICH has requirements from Q1A, Q2 on method validations, impurities in Q3, A through E. You can see all of these guidelines on the left. And if you really want a good primer into getting a drug ready for commercial, reading through the ICH guidelines will give you a very clear understanding. Now, navigating when you need to make some of these transitions and is really where choosing a correct partner or having a strong regulatory team is really important. To navigate this transition from early phase to late phase, the FDA has some specific guidelines. I've highlighted just a couple. There are dozens of guidelines for CMC that the FDA has published. One of the key ones is INDs for phase 2 and phase 3 clinical studies. CMC, what you need to know, chemistry, manufacturing, and controls. But then there's also some procedures if you're dealing with a biologic, and then validation of chromatographic methods. The goals for a late phase transition are comprehensive. We're looking to be able to get ready for commercialization of this drug. We need to ensure we have well-characterized starting materials, intermediates used in the process, and then the API and the drug product. And that's a really key area, and we're going to dive into that in a little bit. Analytical methods need to be validated for their intended use, and they should be appropriately developed. We have to have reference standards that are available across the production process. And this is a big change and transition that happens. All these reference standards are going to be used for our phase 3 analytical method validations. And some of these reference standards are going to be used for routine analysis in QC for our phase 3 pivotal batches, as well as our commercial launch. Specifications need to be appropriate to control all of the critical quality attributes, and our methods need to be robust to ensure that our QC teams can perform this testing long term. So to start out, we're going to talk a little bit about registration starting materials. Registration starting materials, this defines the start of GMP production. And I've got a schematic on the next slide that will illustrate this in further detail. But the selection of registration starting materials is an important transition in phase 3 to get a hold of, because regulators will highly scrutinize your selection of registration starting materials. ICH Q7 and Q11 define a registration starting material as an API starting material. It's a raw material, an intermediate, or an API that's used in the production of an API and is incorporated as a significant structural fragment. And you'll see that on the next slide for what this looks like. It's really important to define this clearly, because a lot of times we deal with very complex early phase registration starting materials, and then we end up getting pushback and have to develop more and more analytical methods. Oftentimes, as Q11 defines, commodity chemicals are generally accepted, and there's a reason for that. As you go into commercial launch, you want to make sure you have accessibility to your registration starting materials so you don't disrupt supply of commercial drug. A registration starting material should be far enough back in your manufacturing process, the selection of what is your starting material, that you can ensure that any impurities that originate in your registration starting material will be purged from the process. So this schematic here just shows an example of a synthetic path for a drug where each of the colors highlight a different registration starting material. And what you can see here is that the active pharmaceutical ingredient, which is made through multi-step synthesis, is a combination of many different registration starting materials. As we move into late phase, we're going to be scrutinizing not only the selection of these registration starting materials per ICHQ-11, but ensuring that we have adequate analytical controls in place to ensure that we can see the purity and impurities of those starting materials, and that the impurities can successfully purge as we move through our manufacturing process. In early phase, when we look at registration starting materials, we will always qualify our vendors. And usually we'll qualify our vendors, and for registration starting materials, we'll have minimal additional testing. So oftentimes when the starting material comes in for a production campaign, for early phase, it'll be tested by appearance. We may run an ID test like FTIR, and then we'll usually adopt our qualified vendor C of A to test that material. For some really troublesome materials in early phase, maybe a purity method would be added, but it's usually not required until you get into later phases. As you get into late phase, your starting materials require extensive testing and justification. You need to ensure you're purging impurities. So we do the appearance in the FTIR, but we also add assay and impurities methods when they're appropriate. And then additional attributes. This is not testing a full API. We're really testing starting materials. So the additional attributes like solvent content in that starting material or Carl Fisher, you would only add those into your manufacturing process or into your critical quality attributes as a specification if they impacted the quality of the production process or the API. So usually we're going to be focusing on trying to make sure we know the potency of those starting materials and the impurity profile, and we've identified it properly. Development in late phase for registration starting materials is very common. And all of these methods, as we transition to the late phase, will undergo a full ICHQ-2 validation. So here's a case study where we have a registration starting material method that needs optimized. Now, this was a method that was brought to us by our vendor. And the vendor method had poor resolution. So what you can see here is I've labeled the peak at about 7 minutes as the registration starting material. This is a specificity mixture that shows the two impurities with the registration starting material. And there's poor resolution between the compounds. We also have what you can't see here is poor sensitivity for the starting material at LOQ. It was barely passing, right about 10-to-1 signal to noise. And so as this method transitioned from early phase to late phase, this method was brought from the vendor to our facility, and we performed late phase optimization and validation for this method. One of the first things that we did is we shallowed the gradient down for a greater separation. So this is just tuning the method down. We changed the mobile phase additive. This particular method used trifluoroacetic acid that has a strong UV response, and that was impacting our sensitivity limits. So we changed to phosphoric acid to eliminate that response, and it gave us a significant improvement in our overall sensitivity. And then we adjusted the wavelength from 270, which the original method had, down to 210 nanometers, and that improved our sensitivity for one of the impurities, which had a very large response factor at the higher wavelength. When we look at this method at the end, we developed a robust method that separates all of our registration starting material and its impurities well. We had a 10-fold improvement in sensitivity. We went to a 95-to-1 at LOQ. We improved our resolution of all of the peaks. So some of the conclusions from this simple case study here is to be very mindful of buffer additives, UV response. Oftentimes, that can really be impacted. It is a minor change that you have to do. Simple gradient changes should always be checked as you're looking at transitioning starting material methods into late phase. And then make sure that you collect photodiode array data for all of your compounds, so you know that you're looking at the right wavelength, so you don't have very large relative response factors, or you may be missing an impurity or have difficulty achieving the right sensitivity at the LOQ. The selection of starting materials, as we dig a little bit deeper, in early phase, APIs are often produced using a customized starting material. And so this illustration on the right for the synthesis of camptothecin shows the illustration pretty well. So we have our API down on the bottom here, and this is a multi-step synthesis. And in early phase, you may select an intermediate of this multi-step synthesis. This one may be 10, 15 steps. You may select a starting material for your early phase. That's a fairly sophisticated structure. In this case, it has a chiral center. It's a fairly large molecule, and it's a big portion of the molecule. What's important to know is that ICHQ11 gives all of the definitions for what a starting material must be. And you have to defend your selection of the starting materials. And in the next case study, you'll see why that's important. If you're not compliant with ICHQ11, what will happen is you'll validate your methods for your customized early phase starting material, and then the FDA will push back and tell you to choose a new starting material and ensure you have methods to control them. It has to be a structural fragment, as I mentioned. You have to have a good control strategy. It has to be commercially available so that it doesn't disrupt supply. And you should have multiple steps so that these impurities can purge through your manufacturing process if there's impurities formed in the initial starting material. So in this case study, we have an API, an active pharmaceutical ingredient, that was completing clinical phase 2. All the milestones for this drug, the efficacy was met. And so as this innovators program is moving forward, they're getting excited into phase to get ready for phase 3 and scale up their production. I think this was about a 5-fold scale in production, so from about 10 to 50 kilos. The early phase manufacture for this program was a one-pot reaction, so just one synthetic step. It was a fairly complex API, but they had one synthetic transformation. They took 2 sophisticated starting materials, they blended them together to form their API. With their scale-up plan, FDA pushed back on their selection of starting materials per ICHQ11, particularly on the point that the API needed to have multiple transformation steps to ensure that impurities can be purged. Also, one of their starting materials had some potential, they had noticed some potential impurities in their API that could have been related to the starting material. And the ICH guideline says that steps that impact the quality of the API must be controlled under GMP. So the strategies that the innovators chose for this program, and there's going to be a different strategy for every type of API. One of the registration starting materials was taken a little bit deeper. They did a deep dive on the starting material, and they fully characterized it and set additional controls and specifications. They also established more reference standards for it. They took a risk-based approach. The FDA may still push back on their approach, but rather than going to a simpler starting material, they felt they could defend having a more sophisticated starting material and but fully characterizing and controlling it. And it's actually a really good assumption to make. If you can demonstrate that this material can be commercially available, you can often get FDA buyout. The impurities in that starting material, however, were fully characterized, and then they took those impurities and converted them to impurities in the API to just ensure that if those impurities ever did crop up in the starting material, they could be seen in the API as well. For the second raw material, this was a 2-pot reaction. They actually took that one a further step back to create 2 new registration starting materials. So that means new methods need to be developed for each set of starting materials, and new specifications need to be established. Both of these new methods were fully validated with assay and impurities methods. And I'm going to show one example of this on the following slide. So this was the early phase approach for this method. In early clinical phase, the starting material, represented by the box on the left, the registration starting material specifications were appearance, FTIR, and vendor C of A. And when you look at the early phase API method, we could see both of the starting materials, as Tracy alluded to earlier. So we can clearly see our starting materials. We have the API. They had done full force degradation, so we knew the method was stability indicating, and this was an appropriate early phase starting material method. As this program transitioned to late phase, for starting material 1, what ended up happening, as shown on the left, is the method for starting material 1. All 5 of these new impurities, which were identified as potential critical quality attributes of the API, were identified, reference standards were made for them, and the method was fully validated for the accuracy of these impurities. But in addition, because this starting material 1 was a sophisticated starting material, they took an extra step and took each of these impurities and transformed them into impurities in the API. And so each of these is a transformed impurity in the API, and then the method was additionally bridged and validated to be able to support not only starting material 1, 2, but also all of the potential impurities that could come from that starting material. And that method was fully validated for that as well. So they have 2 controls, one in the starting material and one in the final API. And this really brings to light the control of reference standards. In late phase, what we find is significantly more reference standards are needed. And this can become a big bottleneck as programs are transitioning rapidly. We often see clients wanting to move from phase 1 to phase 3 very quickly, or have a very, or be focused on a phase 1 approach in the latest parts of phase 2, and then they're working to adapt and get reference standards. For this program, we started the program out with 1 early phase reference standard, and that was the active pharmaceutical ingredient that was used for the potency assay related substance method. As this program transitioned into late phase, the API had 7 impurities, and you can see the starting material went from a control by vendor C of A to 6 impurities that need to be controlled as qualified reference standards. And then as you transition through this for starting material 2, which now became an intermediate, as it got pushed back to form 2 new starting materials, 3 reference standards were required for it, so on and so on. So this program transitioned from having 1 primary reference standard to over 20 reference standards that are needed for late phase control. Again, each of these reference standards needs to be qualified for its intended use, usually by generating a certificate analysis that demonstrates a correction factor, a purity factor. So reference standard management is really a key issue as you transition to the late phase. There are some guidelines that define reference standards and the management of reference standards, and I've highlighted them here. Q7A discusses key aspects of reference standards, and then the World Health Organization has a great guideline on how to establish, maintain, and distribute reference standards. But what happens in late phases, there's this rapid increase in the need for reference standards. And oftentimes this need is paralleling critical timelines for production of phase 3 material. So it's really important if you're an innovator company to have a robust, comprehensive solution to be able to address the complexity that comes as you move into late phase. So if you're an innovator, hopefully you have an internal system that can manage the growth in reference standards as you transition into late phase. If you're outsourcing that work to a CDMO like Cambrex, having a fully integrated service that can identify the impurities, perform structure elucidation, provide synthesis and resupply of these reference standards. You need to onboard all of the methods so that you can qualify the reference standards at the initial. And then being able to manage all of the inventory around those standards. Many of these standards are going to last the life cycle of the drug and are going to be moved to your multiple vendors for drug substance production. So the volume of reference standards, as we look at this in a typical case, in early phase, we start out this is just a clinical development timeline here. In clinical phase 1, we start out usually with 1 to 2 reference standards, maybe an impurity cropped up, or maybe there was a need for something beyond the API to specify. As we move into late phase 2, we're generating additional reference standards, maybe for a starting material method or an intermediate, an IPC control. As the program transitions into late phase, the number of reference standards will grow significantly. A typical phase 3 program it'll typically have 25 to 30 reference standards that range from the API itself to intermediates, all of the impurities throughout that manufacturing process, and all of the starting materials. So at phase 3, innovators are often struggling with that supply and distribution and qualification and requalification. And you're trying to do multiple things in parallel because you need to perform your phase 3 validations prior to your validation campaign. And those reference standards are often needed for some quality by design studies by the chemical development team. So if they're going to be doing a feed and purge study to try to see how these impurities are purging throughout the process, they're going to need to have a handle on, well, they're going to actually need physical material reference standards to be able to spike into their process and see if those impurities are purging out. And as I mentioned, a lot of these standards will get integrated into the final test method. Relative response factors is another important point as you develop reference standards. Because we're going to be validating these methods, we're going to do accuracy and relative response factor determination, not just for the API, but for starting materials and intermediates as well. Because starting materials are simpler components, they're usually smaller fragments of the API, some of the impurities in starting materials can have wildly different relative response factors compared to that starting material itself. So it's really important as you're developing your starting material methods to validate those methods for relative response factor as compared to just doing an area percent measurement. So you ensure that you know the actual weight percent of that impurity in that starting material intermediate and certainly in the API. And with that, I want to transition to the third topic, which is to look at a case study, for the need to develop a robust method. So we talk about robustness a lot as we move into late phase. And so I'm going to walk through this case study fairly quickly, but this was a pretty comprehensive study. An innovator company came with a late phase analytical assay related substance method that was difficult to use. And I've shown a chromatogram on the bottom. You can see there's 6 impurities in this reference standard, or I mean in this API, and then and all of the impurities are resolved. But the claims from the quality control team, the QC team performing this testing, is this method just continues to fail resolution. And the column that is used for this method is very difficult to source. In some cases, the column would be ordered and it would take over 6 months to get this column. So they have ongoing commercial release and stability studies, or commercial ready release and stability studies going on. They've already made all of their PPQ batches, their validation campaign. And so they're really in a critical spot where they're having a difficult time even being able to run this method. And it really highlights, I think, the need for robustness in analytical methods. When we look at the system suitability on the right, I just want to point into the resolution. And when we look at the peaks, we have the three peaks, peaks 3, 4, and the API, and they're all at critical pair junctures. So each of these peaks has a resolution requirement. Peak 4, right before the API, has a resolution of greater than 1, which is not baseline resolved, but it requires a resolution greater than 1. And then there is a resolution requirement between peaks 3 and peak 4. What's important about this project is that these 2 peaks, peaks 3 and peaks 4, are known degradants of the API. And so they want to be able to quantify each of them individually and make sure that they don't get them mixed up. As we look at this method at the initial onset, this is a standard test method. It's just a C18 column, an L1 column. If you're looking at compendial chapters, it's got a good wavelength at 270. Everything seems pretty standard. The method is ion pairing HPLC, which isn't commonly seen, but is easily approachable with a good method. But one of the notices in this method is what I've highlighted in bold. this method used a potassium phosphate buffer at pH 4.55, and there was a very narrow specified range for this method, and that indicates a potential for a robustness issue. In fact, it takes it a step further. The pKa of potassium phosphate is 2, 7, and 12. And this method is set at 4.55. So what we know from the onset, and just looking at the initial test conditions, is that this method is not actually performed using a buffered mobile phase. The mobile phase is similar to water in its buffering capacity. There is no buffering capacity at 4.55 for phosphate. So this method is going to have some challenges. So when we did a critical assessment for this method, we have a table that was probably about 10 more lines long, but I just wanted to focus on a couple of the key parameters of this method. One is that the mobile phase pH was outside the phosphate buffering capacity. The result of that is that you're not going to be able to reproducibly determine the pH of your solution, and that pH may drift because there is no buffering capacity. So it's going to be really difficult when you prepare that mobile phase to get into that set point very tightly. Be like trying to titrate an acid into water within a 0.05% range. Also, critical pair peaks, when we look at the chromatography of peaks 3, 4, and the API, and you look at the structures of those, each of those had carboxylic acids with pKa's right at the range of about 3.9 to 4.9. So right where that 4.55 pH was set in the method. So you have critical pair peaks that are near the mobile phase pH peak with their pKa values. And that means that these peaks are going to be very sensitive to any changes. And these peaks are already nestled very closely together. So there's a lot of variation in relative response or in RRTs that you would expect in a method like this. Also, peak 4, that peak right prior to the API, had an additional carboxylic acid. So there's even more potential for that peak to ionize. And so we are likely to see some significant changes. So the way we typically approach a late phase transition for an analytical method is there's 3 options. One, you can fully redevelop the method if the method lacks enough robustness for commercial. We don't want to do that because there's a lot of work that goes into that. Now, sometimes that's the right path. You'd have to track all of the impurities as you do this redevelopment. And then you're going to have to perform late phase bridging of all of your historical data from your regulatory filings so that you can be prepared to present that to the regulators and ensure that the quality of material you said before is still the same quality. And so you'd be updating a lot of regulatory filings. The second option is to optimize the method within the changes of 621. USP 621 gives allowable changes, plus or minus, that you can do to methods without fully revalidating them. You could also, and this is highly recommended as well, you can simplify complex methods. So if a method has one troublesome spot as you transition to late phase, you can simplify it. Create a second method that can handle that challenge in a very robust way. And don't report that impurity for this method that has a critical pair or a problem that you can't seem to get robustness to achieve. For this case study, the innovator chose the second option to try to work within the bounds to not change the method too much. So when we looked at this method, there were 4 or 5 parameters that we went into. I'm just highlighting, I just really want to focus on the first 3. They were the most impactful during this study. Column selection. We looked at alternate L1 columns. Now, this is a soft spot in USP 621. L1 columns or C18 columns are not always the same. So you'll have to come up with an approach for your validation strategy or justification. But we wanted to look for selectivity changes amongst different types of L1 columns. Also, column temperature. This is a variable that could potentially impact the overall separation, since molecules near their pKa can be very temperature sensitive. Mobile phase pH, obviously, this is a strong parameter. We're allowed to change it plus or minus 0.2 within USP 621. And because of the pKas of these compounds, there could be some selectivity. And to a very limited extent, looking at the ion pairing agent has the ability to impact the separation, but within 621, USP 621, we're only allowed to change it a little bit. So we think, if anything, it would be very minor. So We did a design of experiments, a multi-factor design to improve the robustness of this method. But what we had to do is we looked at 16 L1 columns, alternative columns. One of the goals of this was not just to improve the separation, but to find a column that was readily available, that could be used for the long-term commercial of the program. Because system suitability continued to fail, we actually established system suitability in each of the conditions we evaluated. These are critical pairs. The precision of the analysis was important. And so as we changed pH, column temperature, ion pairing content in single factor and in multi-factor experiments, we established system suitability across those conditions. And we did that for the lead columns that we identified. Out of the 16 column screen, it was about half of them. We also looked at alternative mobile phase preparation procedure, and this was actually a really key development in this study. The way this mobile phase was prepared, you were taking a concentrated acid, adding it to a solution and trying to hit the right pH, and then diluting that to volume. So we changed that whole paradigm and created a precise titration ability to hit pH 4.55 plus or minus that 0.05 limit. And that really allowed us to be able to perform this study and have reproducibility to evaluate the robustness studies. We also looked across multiple HPLC systems in this robustness. We needed to make sure that this method, as it went commercial, could work on Agilent HPLCs, it could work on Waters HPLCs and others. And we looked at precision across a single type of vendor instrument. And so in this case, we looked at 5 different Waters systems to ensure we have reproducibility in this critical pair. We also evaluated 11 different suitability assessments outside of USP 621. So it's saying, okay, well, this is our bounds that we can operate within. Let's take those bounds a little bit better and see if we can come up with a better method. I'm going to highlight just a couple of the points of this study. The first one is the column screening. This is about half of the columns shown here illustratively. And you can see peaks 3, 4, and the API are labeled. What this column study did very quickly was identified alternative columns. The lead column for this method was the beta basic, shown as the standard method here. And you can see peaks 3, 4, and the API are well resolved. We found 2 alternative columns in the first 8 columns and another 2 in the next. And so you can see, if for one example, the X-Bridge C18 provided equivalent separation. The retention time shift, but relative to retention times did not, and all the peaks were well resolved. So this was a big conclusion from the onset of this study, to be able to find a column that was readily available and didn't require long lead times to source. As we looked at the pH of the mobile phase of this method, we found it strongly influenced retention. All of these peaks, as I mentioned, have pKa's really close to the set point of the pH. So we did this variation of plus or minus 0.2 pH. So this is shown at the top at a slightly lower pH, and then the middle is the nominal method conditions, and then at the bottom is the higher method conditions. And you can clearly see as the pH decreases, particularly this peak 4, the peak that's been centered in between, sandwiched between the 2 peaks, it moves to the left to earlier retention time as the peak becomes ionized. And so this was a key conclusion from this study. We also found in this study that lower temperature had a similar impact to pH. So if we drop the temperature down and the pH of the mobile phase, we could actually start moving this peak to the other side of the chromatogram. Now, this was an exhaustive study where we looked at small iterations, but what you can see here is this peak was really buried right between these two. So any improvements we were going to get are likely to have to change the elution order. By changing 3 factors of this column, we changed the L1 column. We switched to a different C18 phase. So we have the XP Shield RPC18. We switched to a low pH within the confines of USP 621 and a slightly lower temperature also within the confines. You can see that we actually reverse the elution order of the 2 peaks, and all 3 peaks are fully baseline resolved. It's important to mention, though, that as you transition programs into late phase, and if you take this type of approach, the conservative thing, this method had already been fully validated, would be with a multi-factor change like this, you should probably really consider revalidating this method, because while you're changing within the bounds of USP 621, you're changing a lot of parameters And a reversal of elution order is a significant change to the method. And you need to be able to track all of the impurities so that you can do your bridging study. So the results from this case study, as we kind of wrap up that study, is we have 3 new L1 columns that gave us full equivalency using the standard test method. And so we took those 3 extra columns that we identified and we fully validated the method for them. And that was a conservative approach, even though we were within the confines of 621, because we are changing the stationary phase. We also utilized in that method the improved mobile phase preparation so that we could always prepare that method precisely. In addition to that, this robustness study unveiled 3 additional methods that we were able to develop from the data. Each of those methods improved the resolution, but reversed the elution order of peaks 3 and 4. One of those was the one I showed on the previous slide, where we operated within the confines of 621, and that method was fully validated. And the other 2 methods, which were outside USP 621, were developed. One was that we could just simply double the amount of ion pairing agent, and we could fully resolve the peak. If we even dropped the pH lower than 4.3 and we went down to 4.2, we started to get outside of one of the pKs of the compounds, and the method stabilized, and we were able to reverse the elution order and completely separate the peaks. Due to the stage of the clinical program, the innovator for this program chose to commercialize adding the additional columns and keeping the method as it is without changing elution order. But what we did is we validated each of these methods. They did adopt all 3 columns, and they adopted the mobile phase preparation. But I think what this robustness study really highlights is there's a strong need to develop robustness into the method. In this case, we proved robustness, that we could make a method robust, or at least precise. Whether that method is really rugged or not is questionable, but we could produce it every time. But we had to work within the confines of a fully developed method that was already in late phase commercial ready state. And so we were able to tweak the conditions and come up with something that works, but developing that robustness into the method is a better design. So to summarize our talk today, in early phase, the goals are very different than the goals in late phase. In early phase, methods need to be scientifically sound. As Tracy mentioned, we don't need to do too much, and we don't want to do too much. As we transition a program into late phase, we need to take phase-appropriate approaches across that journey. The analytical testing strategy needs to be approved by regulators throughout that. As that program transitions into late phase development, we're going to be focusing on compliance to ICHQ 11, and we're going to be leveraging the FDA's guideline documents to help us make that transition. But really in late phase, we're focused on critical quality attributes, whether they're from a starting material, an intermediate, or the API, that we have fully characterized them to ensure we have quality material. And it's really critical during this transition to understand starting materials, that they're justifiable and that they're very well characterized. It's also important to know that there's a lot of growing reference standards. The number of reference standards grows significantly, and we need methods that are designed to be robust. And we can do our best to validate them for robustness at the end, but designing them to be robust is the best strategy. And with that, thank everybody.
Yeah, thank you both for that great presentation. A few people have already submitted questions, so let's jump right into those. The first one is, can you explain the difference between an early stage method qualification and a later stage validation? Who wants to take that?
Yeah, I can take that, I suppose. So there isn't a whole lot of difference. The difference is going to be in how in-depth your precision is between a couple of analysts and different methods, and the performance of robustness. So typically in a qualification, robustness isn't really examined, whereas at the later phase in a validation stage, robustness would be.
Yeah, anything to add, Daniel?
Yeah, no, I think the intermediate precision and robustness are our main focus areas in early stage to late phase.
Great. So let's go straight to the second question then. Regarding starting and raw materials, when do the vendors need to be qualified?
So again, I think Daniel touched on this a little bit in his presentation, but we do qualify the vendors at early stage. What is different is what kind of conformational testing we do at which level. At the very early phases, we're just looking at a confirmation of identity, whereas at a later phase, you want to fully test and characterize that material.
Thank you. A third one is, what proportion of programs do you see that get a pushback in selection of registered starting materials, and when does this happen?
Yeah, so much is going to depend on the chemistry of the new chemical entity. We work with a lot of clients transitioning from phase 2 to phase 3 every year. Probably about half of the programs that we work on will have pushback on at least one registration starting material selection to move it back into earlier stages. This is usually, the feedback for this is usually going to happen at end of phase 2 meetings with the FDA. But there's a lot of opportunities for innovators to be able to find time to meet with the FDA, type A, type B, and type C meetings, and get that initial feedback on whether their selection of starting materials is appropriate.
Great. And the final question, in the reference standards section, I assume many of these late phase standards will actually be retention time markers rather than quantitative standards. Can you comment on this? And who wants to take that one?
Yeah, I can take that one. When a program transitions, like the example that was given earlier, we had a project where we started with one reference standard and we moved to about 22 reference standards. All of those reference standards in that program were fully quantitative reference standards with full mass balance on the certificate of analysis. The reason why that was done that way is that those standards needed to be used to determine relative response factors for the validation. Now, addressing the question, would we have a lot of qualitative standards? The answer is yes. Eventually, after response factors are determined for those methods, many of those standards will transfer from a quantitative standard to a qualitative standard. And so the expectation would probably be about 2/3 of the standards as you go commercial will become qualitative retention time markers rather than quantitative. But you'll need them quantitative at the initial stages.
Right. Did you want to come in on that, Tracy, at all?
Nope, I think Daniel hit it on the head.
Great. Well, and on that note, we've run out of time. So I'd like to thank our speakers, Tracy Milburn and Daniel Kirschner, for that great presentation, and our sponsor, Cambrex, for making this event possible. On behalf of Cambrex and Sightline, have a productive remainder of the day, and thank you for watching.